What if I told you that the alcohol foam dispenser is sitting right outside a hospital room?
Oh, right. The one literally every doctor and nurse is trained to use constantly.
Yeah, exactly. That exact one.
Yeah.
What if I told you it is completely useless against one of the deadliest bacteria inside that room?
It's terrifying to think about.
It really is. Today, we are looking at a bug that uses modern medicine's greatest weapons, from broad spectrum antibiotics to standard hospital hygiene, and it uses them entirely against us.
Yeah, it's honestly one of the most profound paradoxes we face in modern healthcare today. We're so conditioned to think of medical treatments and sanitation protocol as the ultimate solutions, right?
Right.
So to realize they can actually be the catalyst for a life-threatening illness, it's a really tough pill to swallow.
Welcome to this deep dive into our sources on Clostridioides difficile, or as you probably hear it called in the medical world, C. diff.
Yes, C. diff is what everyone says.
Right. So our mission today is to unpack the mechanics, the deeply surprising history, and, well, the bizarre biology of this antibiotic-associated killer. Because C. diff is like the ultimate opportunist.
Oh, totally.
It thrives precisely when modern medicine tries to wipe out other bacteria.
Yeah, and this isn't just a simple story about a dangerous pathogen. If we look closely at the source material, it's really a story about complex internal ecosystems. It's about decades of mistaken identities in the lab, and it ultimately explains why a bug that we once thought was, completely harmless is now at the center of this massive medical crisis.
Right. So let's actually start with that mistaken identity, because before we can even understand how C. diff dismantles the human gut, we have to look at how the medical community completely misjudged this bacterium for what, over 40 years?
Yeah, over 4 decades. To see where we went wrong, we have to go all the way back to 1935.
Okay, 1935.
Right. There were these two researchers, Ivan Hall and Elizabeth O'Toole, working out of the University of Chicago, and they were doing this broad study on the intestinal flora of newborns.
Just general newborn gut bacteria.
Exactly. And during their research, they cultured this strange, completely new, rod-shaped bacterium from the stool of the infants.
Wait, hold on. That doesn't make sense. We know C. diff today as this notorious, often fatal hospital bug, right?
Right.
Why on earth were they finding it in newborn babies? Were these infants, like, showing signs of infection or something?
Not at all. The infants were perfectly healthy.
Really?
Yeah. Hall and O'Toole found this organism in a large majority of the healthy babies they tested. And, because the babies were fine, the researchers considered the bacterium entirely harmless.
Wow.
So they officially named it Bacillus difficile.
Which the sources mentioned that name didn't mean what we might assume it means today.
Oh, right, yeah.
Like, they called it the difficult bacillus, not because it was, difficult to cure or dangerous to humans. It was purely because it was a finicky anaerobe. Like, it grew incredibly slowly in the lab. It hated oxygen. And it was a technical headache for them to work with.
Exactly. It was essentially just an annoying quirk of neonatal microbiology.
That is so funny.
I know, right? And for over 4 decades, it just sat in utter obscurity. It was classified in medical textbooks as normal, harmless flora. Literally nobody was looking at it as a threat.
So then what happened in adult patients that suddenly turned this harmless baby bug into a monster?
Well, the turning point hit in the 1970s. Clinicians started noticing this mysterious fatal colitis, which is, severe inflammation of the colon appearing in patients who were taking the antibiotic clindamycin.
Oh, okay.
Yeah, and these patients were developing severe watery diarrhea, and their colons were coated in these really distinctive yellow-white raised plaques, which are called pseudomembranes.
But they didn't immediately suspect our difficult bacillus, did they?
They didn't. Initially, the dominant hypothesis was that Staphylococcus aureus was to blame for this colitis.
Right. Staph.
Yeah. The actual breakthrough didn't come until 1978 from a research team at Tufts New England Medical Center. This was led by John Bartlett, Andrew Onderdonk, and Sherwood Gorbach.
And they used hamsters, right?
Yes. They set up a hamster model to study the disease. When they gave the hamsters clindamycin, it reliably produced this fatal colitis.
Wow.
Then they took the stool filtrate from the sick hamsters and exposed it to cultured cells in a petri dish.
And they found it was highly toxic to those cells. The sources call it a cytotoxin, which that literally just means a toxin that kills cells, right?
Yeah, exactly. Cyto meaning cell, toxin meaning poison. The filtrate was just destroying the cells in the lab, so they meticulously tracked that cytotoxin back to its source.
And it was C. diff.
It was. The producer of that fatal toxin was C. diff. They published a landmark paper in the New England Journal of Medicine definitively proving that the harmless infant bug from 1935 was actually a major cause of hospital-acquired death.
That is just a staggering reversal.
It really is.
It really shows how dangerous it can be to, prematurely label any organism as just normal flora without understanding its full potential.
Absolutely.
And the identity crisis didn't even stop in the 70s, did it?
Oh, no, it kept going. For 80 years, this bacterium was classified in the genus Clostridium. That placed it right alongside the bacteria that caused tetanus and botulism.
Okay, heavy hitters.
Right. But in 2016, a team led by Paul Lawson used whole genome sequencing to take a closer look at its DNA. And they proved it was phylogenetically distinct. It wasn't a classic Clostridium at all.
Wow.
It had its own entirely separate evolutionary lineage.
So they changed the name to Clostridioides difficile, which that basically just means Clostridium-like, right?
Yeah, exactly. Clostridium-like.
Which is convenient because it allowed the medical community to keep the C. diff acronym that everyone already knew.
Yeah, a happy accident for all the textbooks.
Right. But it really highlights that this bug plays by its own rules.
For sure. And when you step back and look at that whole timeline, it forces a massive biological question, How does an organism that can sit completely harmlessly in an infant's gut suddenly become a fatal pathogen in an adult?
Which brings us to the actual ecology of the human gut. Because knowing C. diff is deadly is one thing, but how does it actually manage to take over the colon?
Right.
And this is where we get into the profound irony of the very drugs meant to heal us.
Yeah, we have to talk about a concept called colonization resistance.
Okay.
When you picture a healthy human colon, you shouldn't picture an empty, hollow tube. It is a bustling, incredibly dense, hyper-competitive ecosystem. It houses hundreds of different bacterial species, and they are all locked in this fierce, constant battle for nutrients and physical attachment sites along the wall of your gut.
I really like to picture the healthy microbiome as this fiercely competitive real estate market.
Oh, that's a great way to think about it.
Yeah, so C. diff is basically a squatter trying to move in, but it just can't get a foothold in a bustling, fully occupied neighborhood.
Right.
Every house is taken, there's no free food, and the locals are actively keeping it out.
Exactly. But then a patient is prescribed a broad-spectrum antibiotic.
Right.
Drugs like clindamycin, fluoroquinolones, or, third-generation cephalosporins. These drugs don't just target the specific infection in a patient's ear or lungs.
Right, they aren't precision weapons.
No, not at all. They act like ecological bulldozers. They flatten huge swaths of the competing flora in the gut. They completely clear the field.
They bulldoze the entire neighborhood.
Yeah.
And C. diff survives the purge because it has a built-in bomb shelter.
Exactly the spore. C. diff forms dormant spores that are remarkably tough. I mean, they're practically indestructible by normal biological standards.
Oh.
Yeah, a thick protein coat allows them to resist high heat, resist drying out on surfaces for months, and easily survive the intense acid of the human stomach when swallowed.
Okay, so this is where we circle back to that terrifying detail about hospital hygiene from the beginning.
Yes.
The sources really emphasize that alcohol-based hand sanitizer does absolutely nothing to kill C. diff spores.
Nothing at all. And this is a massive issue in infection control.
Yeah.
We've built modern medical hygiene around the convenience of the alcohol gel dispenser, right?
Oh, yeah, they're everywhere.
But alcohol only destroys vegetative bacterial cells, the active growing ones. It simply cannot penetrate or damage the dormant spore coat of C. diff.
Meaning, a nurse or a doctor could diligently scrub their hands with alcohol sanitizer between every single patient, doing exactly what they were trained to do to stop disease.
Right.
And still act as a carrier, just transferring viable spores from room to room.
Yeah, it acts as a Trojan horse. The spore just sits on their hands, completely unbothered.
That is so unsettling.
It really is. And this is why strict hospital protocols for known C. diff cases specifically mandate physical mechanical removal. Medical staff have to use soap and water, creating actual friction to literally lift the spores off the skin and wash them down the drain.
Wow.
And for cleaning the room itself, standard hospital cleaners fail. You have to use diluted bleach.
Okay, so let's trace the path here. The spore survives the hand sanitizer. It survives being swallowed and passing through the stomach acid, and it finally reaches the colon.
Right.
The antibiotics have bulldozed the neighborhood, so the squatter finally has space. The spore germinates into an active cell. What is its actual weapon of choice?
Well, This brings us to a fascinating distinction in how we classify pathogens. C. diff operates very differently than many of the bugs we typically think of as invasive.
How so?
It doesn't actually invade your tissue. It doesn't burrow into your intestinal wall or, enter your bloodstream. It literally just sits in the gut lumen, which is the open space inside the colon.
Wait, if it's just hanging out in the open space of the lumen, how on earth is it causing severe pseudomembranous colitis? Like, how does it do all that fatal damage without ever actually crossing the border into the body's tissues?
By launching chemical weapons across that border.
Oh, wow.
Yeah. Once the spores germinate into active cells, these toxigenic strains start secreting two massive protein toxins, toxin A, or TcdA, and toxin B, TcdB.
Yeah.
And these act as glucosyl transferases.
Whoa, glucosyl what? Let's break that down for the listener. What are these toxins actually doing when they hit the cells lining our gut?
Sure, think of it like biochemical sabotage. These toxins enter the colonic epithelial cell, so the cells making up the lining of your gut.
Right.
Once inside, they attach a sugar molecule to a very specific family of proteins called Rho GTpases.
Okay, and what is the normal job of a Rho GTpase in a healthy cell?
They act as the master switches for the cell's internal scaffolding, which we call the actin cytoskeleton. They literally control the physical structure and shape of the cell.
Okay, I'm with you.
So when toxins A and B attach to that sugar molecule, they essentially jam those master switches in the off position.
Oh, so the internal scaffolding of the human cell just completely collapses.
Exactly. The structure falls apart entirely. The cells lose their shape, they round up, and they begin to die. And critically, this collapse breaks apart the tight junctions.
Right, the tight junctions being the molecular glue that holds all the neighboring cells together to form a waterproof barrier, basically.
Yes. And once that glue dissolves, the epithelial barrier is breached. Fluid starts leaking into the colon, causing profuse, watery diarrhea.
Sounds awful.
It is. Meanwhile, your immune system detects the breach and absolutely panics. It triggers a massive inflammatory response. White blood cells, specifically neutrophils, rush to the site of the damage and begin piling up in the tissue.
And that massive pileup of dead cells, fibrin, and panicked immune cells, that's what creates those yellow-white plaques we mentioned earlier, right? The pseudomembranes.
Exactly. The devastation is almost entirely toxin-mediated. The bacteria itself just sits safely in the lumen, churning out toxins that methodically dismantle your colon from the inside out.
That is terrifying.
Yeah.
And just to quickly clarify for anyone listening who might be, piecing together different medical terms, the sources point out this is entirely different from a similarly named bug, Clostridium perfringens.
Yes, very different. Clostridioides perfringens is another spore-forming bacterium, but it usually causes brief, acute food poisoning via a completely different mechanism.
Okay.
It uses a heat-stable enterotoxin. It's almost always a short, self-limiting illness. C. diff, on the other hand, is a totally different beast, deeply tied to antibiotic disruption and prolonged hospital stays.
Right, so the standard toxins A and B are obviously devastating enough on their own, but the sources highlight what happens when this bacterium mutates, when it figures out how to, supercharge that toxin production.
Yeah, that is when you get an epidemic on a terrifying scale. Starting around 2002, hospitals in Quebec, Canada, suddenly saw their C. diff infection rates and mortality rates climb sharply.
And we are talking about the emergence of ribotype 027 here.
Yes, also known as NAP1BI in some classification systems. And just to define that term for a second, a ribotype is just a way microbiologists classify and identify specific strains of bacteria based on their genetic fingerprint.
Specifically the ribosomal RNA, right?
Exactly. And this particular ribotype didn't just stay in Quebec. It caused a massive outbreak at Stoke Mandeville Hospital in the UK between 2003 and 2005.
Wow.
Yeah, it was linked to over 30 deaths and actually triggered a national public inquiry.
So what specifically made ribotype 027 so much deadlier than the standard strains?
Researchers found the answer buried in its genetics. Papers published in the New England Journal of Medicine in 2005 revealed that this hypervirulent strain had a partial deletion in a specific gene called TcDC.
Okay, let's use an analogy here for you listening. In a normal C. diff bacterium, that TcDC gene acts as the natural brake pedal for toxin production, right? It keeps the output of toxins A and B regulated. But in ribotype 027,
That genetic deletion essentially severed the brake line.
Oh, wow.
Yeah, without a functioning TcDC gene, this strain is just mashing the accelerator, massively overproducing toxins A and B.
Just flooding the colon with it.
Exactly. And on top of that, it produces a third weapon called binary toxin, which attacks the cell's actin directly, making it even easier for the bacterium to adhere to the gut wall.
The sheer scale of the damage is just, it's hard to wrap your head around. A major 2015 study by the CDC estimated that in a single year in the U.S., there were roughly 453,000 cases of C. diff, resulting in about 29,000 deaths within 30 days of diagnosis.
It's staggering. By those metrics, C. diff is deadlier than many of the famous superbugs we hear about constantly in the news.
Right, for sure.
It firmly cements it among the most consequential healthcare-associated pathogens in the entire world.
So with hundreds of thousands of people fighting this infection, the obvious question is, how do we treat it? And here we run straight into the ultimate medical paradox.
Yeah.
You are trying to treat a disease that was caused by antibiotics with more antibiotics.
It sounds totally counterintuitive, I know.
Right.
And for about 30 years, the default treatment was an antibiotic called metronidazole.
But the sources know metronidazole eventually fell from grace as the primary weapon.
It did. A pivotal 2007 clinical trial led by Lynn Czar found that for severe cases of C. diff, a different antibiotic, vancomycin, was vastly superior.
Oh, really?
Yeah. Vancomycin cured about 97% of severe cases in the trial, while metronidazole only cured about 76%.
That's a huge difference.
Massive. That gap in clinical efficacy led to a total shift in guidelines. So today, first-line treatments are vancomycin and a newer drug called fidaxomicin.
Right. Fidaxomicin is particularly interesting because it's designed to be much narrower in its spectrum.
Yes.
Its main selling point is that it kills the C. diff while sparing more of the surrounding healthy microbiome. Like, it's really trying to minimize the collateral damage.
Which is absolutely crucial because even with vancomycin, 20 to 30% of patients relapse.
Really? That high?
Yeah, they get caught in this nightmare recurrence loop. You take vancomycin to kill the C. diff, but because vancomycin is also an antibiotic, you are simultaneously re-damaging the very microbiome that is supposed to keep the C. diff from coming back.
Right. So to combat that loop, researchers developed bezotoximab. This is a monoclonal antibody. And what's fascinating is that it doesn't try to kill the bacteria at all. It just neutralizes toxin B.
Right. It treats the chemical weapon, not the bug itself.
Exactly, buying time for the patient's gut to recover. But the most dramatic breakthrough in treating recurrent C. diff didn't actually come from a new drug, did it?
No, it didn't. The real breakthrough came from a 2013 trial out of the Netherlands, led by Els van Noot and colleagues. They compared treating recurrent C. diff with standard vancomycin against a completely different approach. Fecal microbiota transplants, or FMT.
Literally taking stool from a healthy donor and transplanting it into the sick patient.
Yes. The goal of FMT is to instantly restore that diverse, competitive microbiome. By introducing a healthy community of bacteria, you reestablish colonization resistance.
Right, putting the neighborhood back together.
Exactly. The healthy bacteria start consuming the bile acids and nutrients that the C. diff spores need to germinate and thrive, essentially starving the squatter out.
And the results of this trial were just staggering. FMT had an 81% cure rate after just one infusion. Vancomycin alone, only 31%.
Yeah, the gap was so profound that the Independent Data Safety Monitoring Board actually intervened and halted the trial early.
I want to pause and emphasize this for you listening because it is incredibly rare for a clinical trial to be halted for efficacy.
Very rare.
Usually you hear about trials being stopped early because a new drug is harming people.
Right.
To stop a trial because the new treatment is working so overwhelmingly well that it becomes unethical to keep giving the control group the old standard of care that is virtually unheard of.
It's amazing. It proved a fundamental game-changing concept in medicine. Ecological restoration works vastly better than just dropping more bombs on the squatter. It legitimized the entire field of microbiome science overnight.
So we have these advanced hypervirulent strains, we have these wild ecological treatments like FMT, but despite all this progress, the sources highlight a massive ongoing debate in the medical community about who actually has the disease right now.
Yes, this is the overdiagnosis debate, and it stems from a major shift in how hospitals test for C. diff. In the 2000s, hospitals moved toward ultrasensitive PCR testing.
Right. So let's ground this for the listener, because I think a lot of people assume a positive test is simply a positive test.
Yeah, but it's not that simple.
Imagine a patient comes into the hospital for a routine knee surgery. They don't have an infection, but maybe the pain meds they're on cause some constipation, so a nurse gives them a mild laxative.
Happens all the time.
Right. Later that day, they have diarrhea. The hospital protocol kicks in, and they run a PCR test for C. diff.
And because PCR tests are incredibly sensitive nucleic acid amplification tests, that test might come back positive. But PCR only detects the presence of the gene that produces the toxin. It does not detect the active toxin itself.
Okay, so the patient has the blueprint for the weapon in their gut, but not the active weapon.
Precisely. You could have dormant toxigenic C. diff sitting in your gut, not producing any toxin, not causing any disease. You are merely colonized.
Oh, I see.
Your diarrhea was from the laxative, not the bacteria. But the PCR test flags you as a positive C. diff case.
And critics argue this has led to massive overdiagnosis. Like, we end up inflating hospital infection statistics, putting patients in unnecessary isolation, and worst of all, treating them with powerful antibiotics they don't actually need.
Which might ironically cause a real C. diff infection later by wiping out their healthy flora.
It's wild. So how do we solve this PCR false positive problem?
Well, current guidelines now require a two-step algorithm. You use a highly sensitive test like PCR as a primary screen. But if it's positive, you must confirm it with a second test, an enzyme immunoassay, or EIA, that specifically looks for the active toxin. You need both the genetic potential and the active weapon to justify a clinical diagnosis.
It's all about precision.
Exactly.
And speaking of precision, we need to clear up one final major misconception from the sources. We've talked about C. diff as a hospital-acquired infection this whole time, but that entire paradigm is shifting.
Yeah, the hospital-only mental model is officially outdated. Currently, roughly a third of all C. diff cases are community-acquired. These are people who have not been hospitalized recently.
And what's truly concerning is that sometimes these are young, completely healthy people with absolutely zero recent antibiotic exposure. They're just picking it up in their daily lives.
It is a major paradigm shift. It tells us the organism is circulating in the community, whether through environmental exposure or other undiscovered avenues, It's finding new vulnerabilities and reminding us that we don't fully understand all the ecological levers at play in our own bodies.
So let's bring this big picture into focus. C. diff really sits at the exact intersection of three massive modern medical crises. First, the crisis of antibiotic overuse, as we recognize these life-saving drugs are blunt instruments.
Right. And second, the ongoing challenge of hospital infection control. The fact that standard alcohol sanitizer is useless against spores changes how we have to think about basic hygiene.
And third, the critical load-bearing importance of the human microbiome. Your gut flora isn't just along for the ride. It is a vital active defense system.
Absolutely.
Which leaves us with the final thought from the sources. Something for you to mull over long after this deep dive ends. We often think of an antibiotic prescription as a harmless safety net. Like you get a viral cold, you ask your doctor for an antibiotic just in case, right?
But antibiotics don't work on viruses.
Exactly. And every single time an unnecessary broad-spectrum antibiotic is prescribed, it creates a silent, lingering vulnerability. That quick prescription might not cause any obvious harm today, but weeks later, in a completely different setting, it can leave the door wide open for the difficult bacillus. It really forces us to ask, how do we balance the immediate human desire for a quick fix against the invisible, potentially fatal ecological damage we might be doing to our own internal defenses?
It really requires a total shift in how we view ourselves. We aren't just single organisms, we are walking ecosystems, and we have to start treating our microbiome with the respect it deserves.
We really do. Well, thank you for joining us on this deep dive into the fascinating, terrifying world of C. diff. We hope it gave you some aha moments today. Stay curious, keep asking questions, and we'll catch you next time.